This morning it is my great pleasure to introduce to you Dr. William Fenical. Dr. Fenical is a very distinguished scientist. He's a professor of oceanography at Scripps, and he's director for the Center of Marine Biotechnology and Biomedicine. His research involves marine organic chemistry and marine pharmacology, with a focus on chemical defense mechanisms in marine organisms and the chemistry of marine microorganisms. In particular, Bill has focused on the isolation and identification of chemical materials from marine plants and animals that may have potential pharmacological or agricultural uses. Recently, Bill and his colleagues published a pair of papers documenting the discovery of a novel group of bacteria in deep-sea sediments that have been found to produce molecules with the potential in the treatment of infectious diseases, including cancer. Bill has been the recipient of numerous prestigious awards, including the Silver Medal Award from the International Society of Chemical Ecology, the Pauli Fund Award for Research at the Hawaii Institute of Marine Biology, and the Senior Queen's Fellowship in Marine Science, awarded by the Australian government. He is also listed in American Men in Science and Who's Who in the West. Today, Bill will speak on marine microorganisms, the antibiotic era revisited.
Well, thank you very much, and good morning, everyone. And thank you especially for getting up this early to see me. Not used to that type of response. You know, we never schedule 8 o'clock classes in the University of California. That's just not very popular. While my computer is warming up here, I would like to comment upon the history of our expectations of the ocean. And in one of the areas we've had great expectations is that the ocean will provide for us a number of exciting new resources. And this has been a concept and an expectation and a hope for a very long time. But I want to point out today to you that this is now happening, that the ocean now is yielding the sorts of discoveries and particularly products, and in the medical area, the ocean is expanding very, very quickly to provide for us new opportunities in the treatment of human diseases. One of the areas that I'd like to talk about today is with respect to some of the resources in the ocean that you never see. And these are the tiniest resources. These are the microorganisms that live in the ocean. And I'll tell you a story about some recent research that I think is very optimistic and very much shows that the future of the treatment of human diseases will definitely rely upon the ocean, certainly within the next decade and century. Let me go back, though, first and talk about where medicines came from in a more traditional sense. You probably are aware that molecules in nature provided the foundation of the pharmaceutical industry, but this goes back 3,000 years or more to early primitive societies that began to interact with their own environments, testing and sampling different plants, animal species. The environment around them provided everything for their success and for their survival. And through the centuries, we learned that plants, in particularly diverse rainforest areas, contained molecules that would produce cures and treatments for many, many diseases of those indigenous peoples. Ethnomedicine, ethnobotany, were fields that developed over these 3,000 years and in more contemporary times. Beginning in perhaps about the 17th century, we began to understand that those plants contained very, very specific molecules that could be extracted from the plant sources and then used to treat disease. And so the modern view of the pharmaceutical industry was born. Morphine was discovered in the preparation of called opium, which was used for pain treatment in the 16th, 17th, 18th centuries. Other drugs were discovered. Aspirin occurs naturally in the bark of the willow tree. And a variety of other materials over these last several centuries formed the foundation of the pharmaceutical industry, who then went out and produced purified drugs with a more enlarged sense of what human disease is about and how to treat it. And so the pharmaceutical industry has relied essentially heavily upon natural materials that occur. Over 50% today of the products that you use either are of natural origin, extracted from plants or from microbes, or many of them are synthetic products that come from those natural products. But one of the single most important discoveries in the history of human life is the discovery of penicillin in 1929. Arguably, this is the most significant medical breakthrough ever achieved in the history of medical science. When Alexander Fleming, the gentleman on the right, discovered penicillin from common bread mold, Penicillium notatum, it changed the human life to a course of curing infectious diseases, pneumonia, and many of the diseases that were literally fatal prior to these discoveries. Although the work of Fleming was very significant, the gentleman on the left, Selman Waksman, a Ukrainian immigrant to the US, was largely overlooked, or at least not as prominent in the history of these medical discoveries. Waksman discovered in 1942 actinomycin, the first antibiotic from a new group of microorganisms called the Actinomycetes, which are shown on the slide on the left side. These two groups of microbes, the Actinomycetes you see here on a culture plate, and the fungi, more characteristic of early work from Fleming and others, were at one time considered to be very closely related, curiously enough. We now know that they represent two completely different groups. The fungi and the bacteria are separated, enormously separated, by huge biological differences. But nonetheless, these two single groups of microorganisms have resulted in the discovery of over 120 drugs that we use today. And those drugs are of incredible importance in treating infectious diseases, and I list a few here. Penicillin G, the first material that was ever discovered, and now some 100 derivatives of penicillin. And all of these drugs used to treat cancer, used to treat infectious diseases, used to reduce the potent impact of the immune system. The molecule called cephalosporin and related molecules recently discovered, FK506, are immunosuppressant molecules that are taken as drugs to allow transplantations of basically any organ. Without these drugs, heart transplants, liver, kidney transplant, lung transplants would not be feasible. The impact of these drugs on the daily lives of our daily lives is enormous, absolutely enormous. Well, what's the status of this process today? Where are we going? And what about these big industries that spend billions of dollars to discover these drugs? Well, it's in decline. The reason is, of course, that many of the pharmaceutical industries literally scoured every conceivable microenvironment of the planet, all terrestrial, for these bacteria and fungi from which to discover drugs. At the heyday, $9 billion were spent per year searching literally under every rock throughout the entire world. No part of the world escaped the examination of the pharmaceutical industry. And today, though, industries are abandoning this approach. And why is that? It's because not a single new class of antibiotics has been discovered for over 20 years. And I don't think people understand the problem that creates. And I think we literally have our heads in the sand about the issue of the need to have new antibiotics. Now, why do we need these compounds? You know, you read every day about the incidence of drug resistance. Pathogenic bacteria, especially in the hospitals, are now an extremely dangerous circumstance. reaching an epidemic, clearly an epidemic status over the next 10 years. We need to have an arsenal of drugs that we can use to treat those drug-resistant infectious diseases. There's many, many examples. Tuberculosis, which really generally comes across the border from Mexico, is now drug-resistant. We can't treat it. Staph infections, which are extremely common in hospitals, are almost totally drug-resistant. There is a chance, and a growing chance, that if you have a minor surgical procedure in a hospital, that you will contract a drug-resistant, uncurable infection there. Think about having outpatient surgery. So we need to have a continuing new arsenal to combat these problems. We need to know how to find new antibiotics, we need to know where to look, and we haven't known where to look for over 20 years. And so, in the new changing paradigm in the pharmaceutical industry, this massive multi-billion dollar worldwide industry, they are in trouble. They are looking at different sources of drugs, and they are not finding, especially antibiotics, they are not finding new antibiotics. Well, why didn't they consider the ocean? The ocean is a massive part of our planet. 70% of the surface of the Earth, 90% of the crust of the Earth. The species diversity is enormous. 34 of the 36 phyla of life are marine, whereas only 17 of those basic divisions of life are terrestrial. You know, why were we so terrestrial-centric on everything we did? You know, I think it's understandable because the ocean is fierce and powerful and difficult to control. And scientists in the early parts of the 20th century, when this pharmaceutical industry began, were frightened to undertake something so new and so bold. And so they continued along a course until they ran out of those resources, literally ran out of them. Well, let's look at the ocean and the microbiology of the ocean. You know, we know very little about the microbes that occur in the sea. And thanks to Claude Zobel, the father really of marine microbiology, was a professor here at Scripps. And I understand Jean Zobel might be in the audience somewhere. Hi, Jean. Welcome. You know, it was Claude Zobel who brought the attention of the world community to the fact that the ocean was, in fact, different microbiologically. He continued to stress that marine organisms were different than terrestrial ones, and yet no one believed it. They said, well, you know, bacteria and different microbes, they just flow into the ocean from rivers and streams and rain and runoff. But now we know that the ocean is an exquisite composition of life forms that are an enormous frontier represent an enormous frontier in understanding. These life forms are diverse and huge in scope. 1 milliliter, a 28th of an ounce, contains over 1 million different cells, of which we really know very little. When you talk about sediments, or the equivalent of the soil in the bottom of the sea, there are 10 to the 9th, 1 billion different cells in a cubic centimeter, about the size of a cube of sugar. 1 billion different organisms, and yet we know very little about it. Marine science in general is really an unknown science compared to what we know about the land, and it's because we haven't had the long historical interest and interaction with life in the sea. But that's changing. Well, what about the distributions of marine microbes? Of course, they are distributed in very much the same way much life is distributed in the sea. There are tropical places, there are temperate, polar oceans, there are a variety of different habitats where microorganisms live, and we can just simply divide them into those 3 major regions. But when you consider the 3-dimensionality of the ocean, the microorganisms that live on the surface of the sea are entirely different from those that are adapted down to 200 or 300 meters depth. And then what about lower and deeper and deeper? As you change to these different habitats, the microbiology changes dramatically. So wow, what does that add to in terms of diversity and potential and understanding? It's just an enormous field of study. Not only are microorganisms just adapted to live in salt water and live on the bottom of the sea, but something that's very unique and very different about the marine environment is that many of these microorganisms choose to live in association with plants and with animals in the ocean. They form symbioses where they have to survive together because of the low nutrients in the ocean in general. And so we see virtually every form of life, whether it be a plant, an animal, an invertebrate animal, or a whale, having an association of microorganisms that are specific and have a role in the survival and adaptation of those animals. A very different adaptation in this liquid, huge liquid environment. And examples are shown here. Here's the surface of a foraminiferan, which is a small marine invertebrate, and you can see bacteria living on the surface, entirely encapsulating the surface, showing here that this is a single species. Remember, there are 10 to the 6 species in water, so how does one single microorganism adapt so exquisitely to this surface of this animal? You can look at the surfaces of plants. Again, exposed to that huge diversity in seawater, but only see 1 or 2 different forms present on the surface. And if you look inside, you see that sponges and some of these other invertebrates, ascidians, harbor microorganisms that have never been seen before in terrestrial environments. And these adaptations are oftentimes very interesting. Here's the flashlight fish, for example, a nocturnal fish that has beneath its eye sac a pouch containing bacteria that are bioluminescent. These bacteria liberate light in bursts, and members of the same species can recognize where they are in the dark coral reefs, and this, of course, increases the success of reproduction in these nocturnal fish. So these adaptations are enormous and significant. Well, one of the important groups, and I mentioned Salmon-Waxman, because of all of these drugs that were discovered in the soil, one of the important groups are these things called Actinomycetes. They're just bacteria, but they're very important bacteria. And it was discussed and debated for many years, and the latest discussion was by a prominent British microbiologist, Goodfellow and Haynes, and they essentially said that these kinds of organisms don't occur in the sea. Up until recent work, there were no authenticated cases of these kinds of bacteria in the ocean, and of course this may be one of the reasons why the pharmaceutical industry chose not to be involved. There were several paradigms that weren't true. First, people said, oh, you can't culture marine microorganisms. Nobody knows how to do that. Well, nobody tried. Secondly, they said things that are important in drug discovery and in pharmaceutical development don't occur there. They were wrong. So you see, certain types of discouraging comments isolated the ocean from the terrestrial environment for more than 60 years. So let me talk a little bit about some of the research activities we're undertaking, and I'll show you some methods, we'll talk about some results, and I'll show you a little bit about how microbes inhabit various parts of the ocean and try to give you a sense of whether or not we will have a new antibiotic era, whether in fact it will be revisited, the enormous successes of the past. So I want to show some activities. We, of course, are ocean-going scientists. We use research vessels, and we work in a variety of places around the world. Most of our work started out in warmer places, simply because we needed to be in the water for long periods of time. You know, when you can choose, then you don't go to the polar ocean. So we've been working for about 15 years in the Bahamas, very hospitable, warm water environment. We used a variety of research vessels, such as Seward-Johnson, and brought together a group of scientists, quite a large number of scientists, often 30 people, with diverse backgrounds in biology, microbiology, chemistry, pharmacology. And together, this group examined the role of the oceans in this medical field. One of the things we did was to start to sample both water and the bottom, sediment samples. We don't really call this soil, but these are sediments, and they resemble soil in some ways, but are completely different in many other ways. And as we began to sample those sediment samples, beginning in only 1 meter of water, and work out to deep water, at first what we found were very large numbers in the greenish color, whatever that color is called, a whole group of terrestrial bacteria, the Actinomycetes, but they were always terrestrial. A tiny, tiny fraction we couldn't understand. We didn't know what they were. And as we went to deeper water, and of course not very deep, 3 to 6 up to 33 meters only, what happened was those terrestrial forms started to diminish. And what started to increase were a whole new group of truly marine bacteria that require salt for growth. They are exquisitely adapted to the ocean, and I want to tell you a little bit about those. Now here's one example in very shallow water, and if you just sample the sediments in these mangrove areas, you get mostly terrestrial things because they really are the land and not the sea. But living here, about 5% of those that we sample are not terrestrial and have never been seen before. And this is an example of this organism that we could not identify at all. We couldn't even predict where it resided in terms of the biology of this class of organisms. It has a very unique gene sequence. I'll say a little bit more about genes because one of the ways that we identify these organisms is by genomic methods, by the use of sequences of their genes to understand how they evolve and how they relate to everything else. And so this particular microorganism was cultured, and when we did so, we found that it produced a series of new antibiotics of a different structure type than seen before. And here's what they are. They look like the chemistry. You're not chemists, and we spend a lot of time with the chemistry, I have to tell you that, but when we finally get the solution, then it's easy to show and it's over. But the chemistry is the important part. The chemistry and the bioactivity and how these chemical compounds affect pathogens in the ocean and elsewhere. And these molecules, called marinones, are active against the drug-resistant bacteria, against Staph aureus, for example, that's resistant to methicillin, which is the penicillin analog that's used today for most, well not most, for a very large number of infectious diseases. It's also active against vancomycin-resistant enterococcus, and vancomycin is the single most potent antibiotic that we have in the arsenal, and this enterococcus is resistant to it and all of the others. And so a target like this, enterococcus create upper respiratory tract infections, pneumonia, and as well as digestive tract infections. So we were impressed. You know, here's a new molecule, a new antibiotic that was pretty easy to find, and we began to say, well, what if we go a little deeper? How can we discover new microbes if we go a little deeper in the ocean? And of course, you know, we can't dive very deep. The ocean's a deep place. And we can't dive. So we decided we would try to devise and utilize some sampling devices that we could control quite easily. And here's one of the first approaches. This is a little device that we made actually from a lake sediment sampler. It's supposed to be used in a couple of feet of water to sample the bottom. It's a little snapper device here. We call it the mud missile. And we put some fins on it, and we connected it to a big line on a spool. and we drop it off a boat. And of course it weighs about 10 pounds, so it's rapidly reaches the bottom, and when it hits the bottom, these jaws snap and grab some of the surface sediments. Very crude. We're not very high tech, but we're getting there. So this little device, which we never thought hardly would work, began to be usable down to around 200 meters, about 600 feet, and that's pretty good. So we got a little tired, though, of pulling it up by hand from 600 feet. It took about 30 minutes to drag it back, 10 pounds. So this was obviously needed to be replaced. When you get these samples to the surface, here's what they look like. The muds from the bottom are intact, and we can just sample that. We take these muds, we bring them back to the laboratory, and then we devise new ways to culture them. The biggest problem has been that life in the ocean doesn't respond to the old-fashioned methods of culture that were designed essentially by Pasteur. And so we have to use new things. We have to use a variety of nutrients. We use krill paste, seaweed meal, crab shell digest, menhaden meal. We use all these nutrients that we think are restricted or more characteristic of the ocean. And then we can grow things. Now what happened was we began to see that organisms, microorganisms, were living on these micro sand grains or little fine grains in the sediments. These are real muds, really. And as we just scattered some of this mud onto plates, we started to see these microorganisms growing right directly off of the sand grains. They live on the sand grains. And as they began to grow, we began to culture them in larger scale, and you see them here growing on an agar plate, typical microbiology type of approach. And these are organisms that looked a lot like a known group called Marinomyces, but turned out to be different, turned out to be very different. We began to see them everywhere, and here's an example of 12 of them, and they are different in appearance from lighter colors, pinkish oranges, to dark black in colors. They produce a variety of pigmentation. They have an obligate requirement for sodium ion, which is an obvious thing that allows them to be adapted to the sea. But they're new, and they're very different. And we began to see that these are an entirely new genus of microbes, which we coined the name Salinospora in response to their saline requirements. We've now amassed more than 3,000 strains of these, and how do we know that these are unique? I mean, obviously, they're from a unique place. We find them down into the deeper parts of the ocean, but how do we know they're truly unique? Well, to do that, we use the technique of gene sequencing, and we use a particular gene called the 16S ribosomal RNA gene, and we sequence the DNA in that gene. And when we do that, we see a comparison of what this organism is composed of at the fundamental genomic level. When we see that comparison, we can compare it to everything that has ever been isolated and understood, because all of that data from the land are in databases that we can computer-wise compare. And this is where we use the new field of bioinformatics. This is an enormous amount of data out there for thousands, maybe hundreds of thousands of terrestrial things. And that comparison allows us to say, these are unique. These have never been seen before. So this represents that gene. This is the size of the gene, and these little white spaces here represent what we call the characteristic signature nucleotides, which means that's how we know for sure that these are different. Nothing has ever been seen that contains these signature nucleotides. And so these are a unique group of microorganisms. Well, where are they? They appear to be mostly tropical. And so we began to look at them around the different parts of the earth. We collected them in Guam, in the Red Sea, in the Caribbean, in Bahamas, in the Sea of Cortez, in Hawaii. And we're pretty confident that they are localized in warmer waters and that they are primarily tropical. We have not seen them here in La Jolla. Now, of course, one of the questions you have right away is, okay, these are organisms, and maybe they're different, and maybe the ocean contains some interesting things. But really, are they going to provide the foundation for the discovery of pharmaceuticals? Do they produce things in the ocean, in the bottom of the sea? And is it the same as the analog microbes on land? And to answer that question, we decided to take 120 of the different Salinospora isolates, grow them, and expose them to a variety of different medical bioassays to look at the potential for the treatment of human disease. We were really shocked at the result, especially in the area of cancer. Over 80% of these microorganisms produced molecules that were inhibitory to colon carcinoma at levels that are well below those required to be a drug. And these are the crude extracts. These aren't purified yet. 80% of these produced molecules of some potential. Over 30% of the culture extracts were active in the antifungal assay against a drug-resistant form of Candida albicans, which is the major pathogen causing mortality in immunocompromised people, HIV or transplantation patients and so on. Over 35% of these culture extracts showed antibacterial activity against our panel, but against vancomycin-resistant enterococcus again, at levels that suggested that they were incredible potential. Furthermore, we looked at them chemically, and what we saw by just a simple method and a beginning method was the presence of molecules in these strains of Salinospora that had never been seen before. And so we began then to be stimulated to spend the much larger amounts of time and money to go into these cultures and find out what's there. So to do that, the first thing we can do is to culture them. And here you see a shake flask cultivation process that we use for growing bacteria. This is common. These are set up in the basement of Scholander Hall with a facility that's temperature controlled. And we inoculate 1 liter in each of these 2.5 liter flasks called Fernbach flasks and grow them in these marine nutrients. When you do so, they grow to high densities, they produce materials, and again, the crude extract of this strain, called 392, showed this potent activity against colon carcinoma. As we began to work up the process, we took the flasks and we separated them out, and this just shows a process by which you separate and purify the active ingredients from these mixtures. And we ended up with a molecule down here, extremely potent anti-cancer drug called Salinosporamide, which captivated our interest for some time, very potent inhibitor of a variety of cancer cells. Here's the structure of this for anybody that's a chemist. What's unique here, of course, is that this is unlike any other molecule that has been described from any microbial source to date. Notice it contains a chlorine atom. It's using chloride from seawater. Why not? There's a lot of chloride in the ocean, and it uses chlorine and puts it into these structures, accentuating their biological activity. Now, I won't say much about this, but this is a panel of 60 different cancer cell lines that are tests run by the National Cancer Institute. And on the right-hand side, this is the LC50, meaning the concentration that is effective at killing cancer cells. And as you move up, the center line is the median activity. So anything to the right shows highly increased ability to kill those cells, and anything to the left shows decreased ability to kill the cells. Now, what's important here is that this molecule shows about a 10 to the 4th increased potency against certain kinds of cancers. And it leaves other cells alone. And that's important. That's the future of cancer chemotherapy, is to have drugs that are selective and non-toxic to human cells. So when we see this kind of selectivity against melanoma, breast cancer, ovarian cancer, among others, for that you can read if you could see this better, it's an exciting result. And it tells us that this molecule has some form of unique kind of biological activity that we don't understand. Well, we found out what it is. And here's an example of the activity. This is an important molecule because it interferes with a thing in the cell called the proteasome. I won't say much about it, but it's an important new target. There are no drugs currently available that affect the intracellular proteasome. So this molecule goes into the cell, seeks out a specific site in the cell, and then inhibits the role of the proteasome, which is to degrade aberrant proteins that are floating around in the cell. Very effective, an IC50 of 1.3 nanomolar, very effective, right in the range for drug discovery. There's another compound called omurolide, and before this was the most potent known molecule that had this effect, and this is about 30 times more effective than omurolide. So it's a compound of considerable interest that is currently being developed here in La Jolla and possibly also by Bristol-Myers Squibb. There's a bunch of other things found in the mixtures, and their chemistry is robust in this material. There are many, many new molecules, and we don't know every biological property, but they're all in here. They're antibiotic, they have anti-cancer activities. This single strain produces about 30 new organic molecules that have bioactivity. Now, one of the interesting things about microbes is that they're very hard to predict. And this shows an effect that was surprising to us. Here, for example, is when you grow this organism for 8 days, here's the major proteasome inhibitor, Salinosporamide A. Here are all the minor ones here. But if you let it grow for 11 days, it does something that's hard to understand. It reabsorbs this molecule, you can see it going down, and produces 2 new compounds here that are unusual molecules that we've never also seen before with very bizarre chemical structures. And I told you we are always working on chemistry. But here they are. They're molecules that have structural features that have never been observed in any chemical structures before. You can see a ring here with, again, this element of chlorine on this position in the one and on this position in the other. They're isomeric materials that were discovered and described by Greg Buchanan in my lab. So here's an example of these very different kinds of structures, something very potent from Salinospora, and molecules of unprecedented structure types. The first study we did of more than 3,000 strains. Okay, but is the ocean that shallow? It's not. It's deep. And how do you get there? And how do we get those muds at the bottom of the ocean? And who cares about these muds? Another question you could ask. Oceanographers have not been entirely fascinated with the bottom of the ocean for a long, long time. There were early studies, but they have to work very hard to get a fish in the picture, because the ocean really, or anything, here's a little crab and a sea pen. The bottom of the ocean is basically a desolate, dark, cold mud place, and throughout, except for the geothermal vents and areas of warm water where there's a lot of interest in the oceanographic community, these areas are not so important. For us, they look like an enormous frontier. And so we've wanted to develop ways to get to those deep oceans and culture deep ocean life, microbes from the sediment. And we decided to do it ourselves and to innovate in a way where we could produce sediments from deeper oceans, but at a very fast rate. One of the problems with the use of a big ship is that it's a very big process. to get a sample from the bottom of the ocean is going to take all the ship's crew, all the people running the ship, people standing on board doing a variety of things, and 6 hours later, you pull up a mud sample. Of course, you have 50, 60 kilos of a mud sample, but we need the amount equivalent to the size of a pea. So how are we going to get those, and how are we going to get them repetitively, routinely, and low cost? Well, we amplified upon some deep-sea fishing reels. What you see here is an Italian deep-sea fishing rig that is used to catch fish in around 2,000 feet of water. And we modified it, first of all, to put this mud missile on it. And we've now been able to retrieve samples using that to about 1,200 meters. It takes 14 minutes to drop to 1,200 meters. And it's not a very dynamic looking sampler, so it doesn't work very well. But we're able now to look at those sediments in 1,200 meters, and they are robust and unique and exciting. And the organisms there are really showing to be interesting. Okay, prototype number 2. Let's streamline this sampling device, and let's try to achieve some other things. So at the Scripps machine shop, we built a miniaturized corer. This is only about 1 inch in diameter at the bottom. And this device then attaches to the reel from the previous photo, except that we've expanded it with some plastic expansions. And now on this reel, we have 3,000 meters of a very high-tech braided nylon or Dacron fiber that's 120 kilo breaking strength. Very, very strong. And this item weighs about 8 pounds, and we can adjust the weight by putting different ballast weights, lead weights on the shaft at the bottom. Now, to be honest with you, we haven't used this yet, but we have a great deal of hope and think that the design that this has been made, the weight distributions are going to allow us to drop this down to 3,000 meters and more. By adding weight and adding this Dacron line, another even 3,000 meters to this Dacron line, we can go quite deep, we believe. We don't know for sure. Incidentally, these are now becoming very high-tech. Working with our Scripps machine shop, we get very high-tech very quickly. These are titanium fins, of course, which are light at the top and give you that resistance in the water so that this mud missile, too, now will proceed very rapidly with no resistance to the bottom. Well, what other things have we done, and how big is this? One of the things we've worked with are some colleagues in the Sea of Cortez down near La Paz at the University of La Paz, and we had an opportunity to use several ships there. One of them is the Golden Shadow, and to then sample in a variety of environments there. And what we found there was another new group called the Marinomyces. Again, a name that we coined and that we believe is an adequate description of these. They're related to the Streptomyces, and they too are producing very, very unique materials. So far we have only 8 strains of these, but every day we find 2 or 3 or 4 more, and soon I think we're going to know how to selectively find these and culture these in these deep sediments. So here's another whole new genus. There hasn't been a new genus in this group of organisms described since 1955. So here's the tree of our life. We look at it as where are we contributing to an understanding of this group of microorganisms, of medically important microorganisms, and all of the things in yellow represent the life forms that are found on land. Those in color, the Salinospora here, Marinomyces, and then other groups, in fact there's several more right over here at this point too, represent new things that come from the ocean. And one of the things I want to point out is that this group of Salinospora, which are so predominant now, they emanate at a point in this tree, radial tree of life, which suggests that these might actually be the predecessors evolutionarily to all of these related organisms that live in the soil. We can date these by changes in their genes. And these organisms, this is about 120 million years ago, where this radiation of diversity in life took place, and this is coincidental with one of the major subsidences of our sea level. And so were these organisms the progenitors, the predecessors of all of these antibiotic producers? Maybe so. You know, we only see a tiny tip of what is a huge iceberg of discovery at this point in time. Our abilities are horrible to culture and isolate these. You know, we've just found them in the last couple of years, and every day there's a new revelation about how to do the work better. And so I think we're looking at something that will expand in an exponential way to show an enormous contribution to medical science. What are the conclusions? Well, if we use different methods, we can find all these organisms in the sea. Forget it that people said they weren't there. They are there, and they are truly marine. They're exciting. We're isolating new genera, the Salinospora, the Marinomyces. These are huge new groups of organisms, and more are on the way. Do these organisms produce antibiotics and anti-tumor drugs? You bet they do. I showed you one example, but we have 10 projects in progress right now where we're isolating molecules we've never seen before, very potent anti-cancer and antibiotic activities. Final question, can the ocean yield the new antibiotics for the next century? I'll leave it to you to think out. I won't stick my neck out, but I'll tell you one thing. If I could buy stock in it, I'd do it. What remains is that I talk about the people who did all of this work and our sources of funding and so on. Paul Jensen is the professional microbiologist who's worked with me for about 12 years, and the others, Chris Kaufman, Tracy Mincer's a student, excellent student in molecular and microbiology. Tatum Neely and Alvin work in helping us to culture things. And many of the people in my lab are chemists, of course. Doesn't look like it by the list, but Greg Buchanan did some of the work here. Rob Felling, who's back in Europe. Charles Patharana and Matt Woolery contributed enormously. And of course, we have bioassays. Sarah Kelly runs our infectious disease assay system here at Scripps. And then we have financial support for both ship operations from National Science Foundation, from National Cancer Institute. Fortunate to have access to funding from the BioStar program, the University of California program, with some matching from Nereus Pharmaceuticals, and then foundation support for ship time and so on. And lastly, this is just to show you, this is the Salinospora Marinomyces team. Paul Jensen leads this team, and I mentioned here's Tracy Mincer, a graduate student, and here's this unique molecule, Salinosporamide A, the first molecule from this very large group of new organisms. Lastly, let me say what a pleasure it is to talk to you. I hope you have some questions. You know, I really am excited about this field. Perhaps you can see that. It's like exploring. I never thought there was anything left to explore on Earth, but you know, you start looking in these sediments and you have a microbiology interest. Wow, you feel like a 14th century explorer, and it's really been exciting. I want to thank you very much for your time. When you look at the deep oceans, you're absolutely right. The pressure in the deep oceans is a considerable pressure, obviously. What we know about that at this point in time is that the pressure requirement doesn't really begin till about 3,000 meters in depth. And we have to overcome that in time to culture organisms under pressure. And that's not really a problem. Professor Yayanos here at Scripps has the capacity to grow bacteria at -1 degree centigrade and at 400 and 500 times the pressure at the surface. Huge applied pressures. And he can grow them in chambers. So it's a technology. Now, we're not focusing on that particular problem now because there's huge amounts of work in shallower water than 3,000 meters. But you're right, long term we would have to probably address that issue. You know, if we collect something, it requires an immediate attention. And without that immediate attention, many things will die right away. So we have to, if we're in a remote area, we have to have some form of laboratory. If we're on a ship, we have to have a laboratory facility available. We can do some things that we can transport and preserve them. We can freeze some samples, we can dry some samples out, and still retrieve viable microbial life. However, the best scenario is immediate processing. It's like collecting a fish. You know, what are you going to do? You better hurry and do something. Well, we think so. What is known about halogenation in general, and chlorination certainly, is that there are enzymes that chlorinate, and those enzymes are available in a variety of sources. Certainly in microbes do chlorination and other things. There are even some chlorination processes in trees. They're very rare, but they're there. The thing is, we don't know about the enzymes, we don't know the process. And when you look at this, it's called a primary chlorine. It's on the end of a chain. And that's something that we don't understand at all. So I think we're going to see some new enzymes, some new genetics, and we are looking through a collaboration at the genetics of the production of these molecules and hoping that in the long term we'll really understand how they differ from terrestrial forms. In the Caldaromyces chloroperoxidase and in the human thyroid, those systems are halogenating activated sites on organic molecules, which you can do with the free halogen, with iodine or chlorine or bromine. It's not a very unusual process, and it's found all throughout lots of different life. What I'm saying is that this position of chlorine is on what's called an unactivated carbon, and we just have no idea how that could happen. We'll have to find out. Professor Space is asking, do we know something about the sedimentology and the relationship of that to the organisms that might live there? And maybe, I guess we could use that to target some diverse areas. So far, what we know about is these are highly calcified sediments. They're mostly tropical. In many of the cases, the muds we pull up from 12 or 1,300 meters are white, indicating that they are, basically skeletal remains of calcified organisms. We know a little bit about that. We've looked at the infauna, the animals living in there, and have seen lots of things, not as much as we anticipated from the literature. It looks a lot less life-rich than we thought it would be. What we know about the organisms we're working on now is they're not associated with any of the life, but they live on the grains of the sediment grains, and they must therefore want to be on surfaces. But that's all we know, and we do need some help in that. We'd like to put together a program where we get geologic understanding and more understanding from some of the deep ocean biologists who have a specialization in the sediments. It can help us a lot. His question has to do with the stratification of the sediments. Of course, we know that in different areas, as we go deeper into the bottom sediments, we turn from an aerobic environment into an anaerobic environment. And when that happens, the microbiology changes dramatically from a diversity of air requiring to organisms that undergo fermentation and don't use oxygen. So it's very important that we know something about these sediments and where to collect them. We haven't had that opportunity with the grabbing device because it only grabs in the top maybe 10 centimeters. Most of that's an aerobic environment. But with the sampling corer tube, that device will collect sediments which are stratified, perfectly stratified and housed within a plastic insert tube inside that we then take out and carefully push out, and it remains stratified. And then we can cut areas of around 1 or 2 millimeters can be cut and examined microbiologically. We haven't done that yet, but of course that's why we built the corer, was to address the issue of stratification. And we're anxious to know where these guys are. Are they on the surface? Are they inside? And other things too affect these sediments, such as nutrient loads in different parts of the sediments. You know, you have a whale drop into that sediment, a whale carcass, and you have a different microbiology. So we're anxious to learn all about that. We have a lot of work to go.